I'm going to present some preliminary data of some new research that we're still in the middle of, sponsored by the Lipedema Foundation. This talk is structured for a general audience, so if you're technically inclined, you're a scientist or a clinician, and you would like more technical details, please connect with me later and we can go into it in as much detail as you want. And one other point that I would like to make is that this experiment is still underway. It is still blinded. So the experiment was done blinded. I do not know which patient is which patient, who is control, and who is lipedema. So we have to wait for the unblinding before I can tell you a full story. For the moment, this is a vignette, a glimpse of the data underway. The experiments are done, but the analysis is still ongoing, and I will tell you why. So nothing to disclose. And this audience, I do not need to point out that there are no proven biomarkers for lipedema. There are no known genetic drivers, although we know that it does run in families. There are no animal model systems that recapitulate lipedema adiposity. Now, there are for obesity. So lipedema is different, but there are no animal models that capture lipedema phenotypes. There are no in vitro models, meaning there are no cell culture systems or tissue engineering systems or ex vivo systems that capture the properties of lipedema. So how do you study such a disorder, which depends entirely on human patients and human patients alone? And obviously, you cannot experiment on humans. You can get samples. So any disorder that for which you don't have model systems, research is proposed very slow and very, very difficult to do. So how do you approach them? So one approach that is current, given the status of technology, is hypothesis agnostic, unbiased, genome scale approaches. So meaning you look at everything that you get hands on through high throughput technologies. And the model here is that if you were doing something hypothesis driven, the idea is, let's say there's an intruder in your house. You're saying, hey, I'm going to look in the basement because there's a door to the outside. Or I heard a noise in the basement. I'm going to send somebody in the basement to look. Your hypothesis is there's an intruder, which is your disease. Something's going on in the basement. So you're looking in one spot. Hypothesis agnostic would be you're going to send somebody to look in every room, look in the yard, look in the garage. You're going to take as global a look as possible and try to get a handle on what may be going on. Is it only one intruder? Is it two? Is it multifactorial? So that is what I mean by hypothesis agnostic, unbiased, genome scale approach. So we're going to look at the molecular landscape at genome scale, and your choices are pretty wide. You can look at the DNA, you can look at the RNA, you can look at the proteins, you can look at the metabolites and metabolomics, you can look at epigenomics, which are genetic modifications of the DNA independent of the DNA sequence. And you can look at ex vivo tissue models and organoids. They don't, ideal models don't exist, but they could if we got some insight. And you can look at a systems approach, multi-scale models, meaning going from molecules through organelles, through cell biology to tissue. And you can do network analysis, which is mathematical analysis of the interactions of all the biochemical pathways that are affecting lipedema. And you could do something called high-content imaging. I'm going to talk to you about high-content imaging today. But the main idea of presenting this slide is to say that the foundation has made awards in all of these areas. And all these experiments are underway, and some of those we will hope to get results in 2025. One of those you will hear in the speaker after me, who's here, Pamela Nonanankam. She's going to talk about transcriptomics, RNA approach. But that is also a hypothesis agnostic, unbiased genome scale approach. So the approach I'm going to talk about is high-content imaging. What do we mean by that? We are using an approach called cell painting. So cell painting, anytime you want to visualize things within the cell, we use antibodies normally. But antibodies are a lot of work, a lot of time and money to generate, and you have to do them one by one. So instead of antibodies, we're using fluorescent dyes. 6 different fluorescent dyes that allow you to visualize DNA and RNA, endoplasmic reticulum, nucleoli, cytoplasmic RNA, mitochondria, which everybody probably knows, generate energy in the cell, and then the cytoskeleton, actin cytoskeleton, Golgi, which is where the proteins are made, and the plasma membrane. You don't have to go into the details of all the organelles. just that you can visualize every major organelle in the cell by using 6 different dyes. And then you do computational image analysis to estimate 3,000 different parameters. So usually when people do low throughput experiments, you look at 1 or 2. You look at how much lipid there is, or how many lipid droplets there are, or is the structure changing. Now with 6 different dyes and computational capacity and very high resolution microscopy, you can estimate 3,000 different parameters for each cell. And you can integrate it with RNA-seq and other global approaches. And eventually, the hope is to perturb these cells and to study the benefit and the effects of those perturbations and drug screens as well. So here is a quick preview of what that looks like. This technology for adipocytes and for lipid cells was developed at the Broad Institute in Boston, which is affiliated with Harvard and MIT. So we took advantage of some technologies they developed to apply it to lipedema. This is one of the missions of the foundation, is to take the latest technologies and adapt them to lipedema. Lipedema is a nascent field. It is new. So we borrow from everywhere that we can and bring new technology to study this disease. So what you see here is, do I have a thing? No. Okay. I'm not sure the pointer is working, but anyway, you can see it. So I just want you to focus on the panel on the left. There are 4 channels that are shown, which is hexed, mitotracker for mitochondria. The yellow tracks the membrane and the cytoskeleton, and the green is the lipid. So that is the lipid within a particular cell. So you have 6 dyes, 4 different channels of color, and you get 3,000 different parameters. And then you can integrate it later on. All this integration with all the other data will happen sometime later this year after we get the preliminary data. The important collaborators for this project are Sarah El-Gadban of the University of Virginia and Bruce Bunnell, from whom we obtained these patient-derived cells for lipedema. Now, what did we do? We looked at lean, obese, and lipedema adipose stem cells obtained from El-Gadban and Bunnell, so they are collaborating with us very closely. We initiated differentiation of these cells to adipocytes. Remember, these adipose stem cells, they're called adipose stem cells, but they're actually mesenchymal cells that can make many, many different lineages. They can make bone, they can make vascular tissue, they can make cartilage, they can make many different tissues. We are using a protocol that makes them turn into adipocytes. And then we're asking, are there ways in which lipedema adipocytes are different from obesity adipocytes or from lean adipocytes? We follow them for 21 days. And then at different time points, at days 8, 14, and 21, we characterize the cells, we image them, and we measure 3,000 different parameters across those 6 different dyes. So you get a very detailed look at the cell. And to tell you how detailed, the minimum number of measurements that we're taking is 2.6 million. So it takes terabytes of data. It'll take a while to analyze. The data analysis is underway, so what I'm going to show you is just a glimpse of where we are. And as I mentioned, the experiment is done, data analysis is underway. So what you want to see here are the 4 channels. So the first one shows you the DNA in the nucleus. The second one shows you the RNA. The third one shows you the skeleton, the cytoskeleton of the cell, which maintains the shape of the cell, helps it move and do a lot of cellular things. And the last one is the mitochondria and some other markers in there. And the last panel, you see lipid. So at the bottom, you see the differentiated cells. Now they're adipocytes. The green signal is the lipid, and the top is undifferentiated. Let me give you a close-up look here. So the first slide on the left is undifferentiated. The next slide is at day 21, differentiated. All the green that you see is the lipid, is the fat content inside every cell. So now, after 21 days, these have turned into adipocytes, and all the different colors are represented. These are merged images where you can see all the colors are imaged. And these are the ones that are being computationally analyzed at different stages. So I'm just going to show you a quick glimpse of 2 data points. This is mean droplet count. How many lipid droplets are there per cell on the average? All I want to get from this slide is that there's no difference between the patients that is significant, too significant, maybe a little differences, but the lipedema adipocytes seem to have as many droplets as do same amount of droplets as do the obese or the control ones, lean controls. And this mean droplet area is the same as well. So it is in what the difference here is not in the amount of adiposity per cell. Looks roughly the same. It is not finished analyzing yet what we're looking at. This is just to give you a rough idea of one parameter out of 3,000 that we will be looking at. So, and this here is what we call a UMAP. You don't have to worry about what a UMAP is. This just means that you have 2.6 million parameters. We reduced it to 2 dimensions. 2.6 million data points reduced to 2 dimensions. And at the bottom left corner, you see these are the differentiated cell lines after they've become adipocytes. And at the top right, those are the cells before differentiation. And then the far away clusters that you see of little, maybe the different patients, I don't yet know who's who. And the distance tells you how different they are from each other. The closer they are alike. The further apart the clusters are, they are different. So that is the goal of the approach, is to ask who is close to whom, who looks alike, who looks different, and then try to get a handle on why are they different, and how can we move them with drugs or with other treatments from one point to another. And so the experiment is complete, as I mentioned to you, and I don't have to go through the slide. I've mentioned everything to you. Where are we going next? The next, we would, once we get the results, we would do more perturbations with drugs, and that can be therapeutic. That would be the ultimate goal, but for the moment, those goals would be more to find perturbation molecules, is to disturb the system in certain ways, and then see how those changes tell us something about the properties of lipedema cells. So a drug screen would be one of the things next. But one point that I thought I would like to leave you with is live cell imaging and time course. There are new technologies available now where you don't need those dyes. When you use those dyes, you're fixing the cells. At that point, the cells are dead. They are attached to the thing. They've been fixed. They're dead. But now you can look at live cells, and that's by a technique called holotomography. So we are talking to a company that looks at holotomography. These are living cells, and all the droplets you see are lipid droplets. And now here is a video of these living cells accumulating the droplets, and you can follow this over time. So the last slide I will show you is here. These slides, if you look at the graphs, you can measuring lipid droplet volume and droplet concentration over 95 hours. So you can see that the lipid is being made. It is increasing slowly, as is the lipid concentration. So from the pilot experiment, we will get a lot of information on what the properties of adipose stem cells are in lipedema, in obese, and in lean. And then we can identify specific time points where important biological things are happening. And then we can use something like holotomography to look at living cells and see those things actually happen. We can see the lipid droplets developing, and then we can perturb them. So this is fixed cell imaging and live cell imaging will complement each other to get a handle on the biology. And here's the contact information. And last but not least, I should thank the organizers for giving me the opportunity to speak. And although I happen to be the speaker standing here, this is a team effort. So the science team included Laura, whom you just saw. It includes people who were all at the conference. Jesse, Stacy, our biobank manager, Elena, who manages all our data, and Stephanie, who manages does all the project management for us. It's a lot of work. Doing many of those things is an extraordinary amount of work. And I just happen to be the speaker up on stage today. So next time I'm here, I hope I'll be able to present real data in a more comprehensive fashion. But it'll take us another month or two to analyze 2.6 million parameters at a minimum. It's actually way more than that. Thank you very much.